Hello friends, today we will do the embryology question. First we will start with 2014's question paper. So our first question is, in the species where pollen matures and is released prior to the maturation and receptivity of the gynoecium, the condition is called. Since pollen is released prior and before gynoecium, so that's why it will be options are protogyny, protoandry, dichogamy or andraci, and our answer will be protoandry, where andrecium is coming first because pro means first and then andry. So here our answer will be number B. Yeah, because pollen is the male gamete is released prior. Okay, next question. A pollen grain with colpi occurring in the equatorial region is called A. Zonocolpate, B. Zonoporate, C. Zonoaperturate and D. Colporate. We have to think first that equatorial region means zono, and then it is saying that it has a colpi. So in the equatorial region there is colpi, so the answer will be zonocolpate, not zonoporate, because its porate is different than colpate. Porate is a pore and colpate is a furrow. Yeah, we have to remember that. Now our answer being A over here. Next question. So growth pattern in the pollen tube is similar to that of fungal hyphae or root hairs in exhibiting A. Oscillated elongation of tube, B. Tip oriented growth, C. Multiple oriented growth, and 4. Predetermined path of growth. Answer will be tip oriented growth. Next question. Pollen tube attraction towards the embryo sac is mediated by small peptides produced from the Synergist, egg cell, central cell, hypostase. The answer is Synergist, which produces some peptides that are used to attract the pollen tube towards the embryo sac. Yeah, answer being A. Next question. Cellular endosperm formation is characterized by, so it has to be a cellular endosperm, and then it is saying that there is absence of free nuclear division. Nuclear divisions are not accompanied by cell wall formation. Primary endosperm nucleus divides to form a large micropyle chamber and a small chalazal chamber, that is halobial. This is nuclear endosperm formation. Then autonomous development of endosperm without fertilization, no, we are not talking about that, because that is apogamy. First of all, this absence of free nuclear division will be the right one over here, that is cellular endosperm. Okay, next question. One of the essential prerequisites for normal microgametogenesis to proceed is, microgametogenesis is formation of male gametes, the process in the male flower. So first option is formation of two sperm cells before the release of the pollen grains. Do we always require the sperm cell to male gametes to form before the pollen grain is released? No, there are plants whose pollen grain after release they get the genetic cell gets divided into two sperm cells, right? So this is not a essential prerequisite. Next, vacuole formation and asymmetric division in the microspores. Well, this is a point which we can consider that this is an essential prerequisite, but let us see the other options. Physical association of the sperm cell with the vegetative nucleus, no, never a requirement we need. Next is the attachment of the stigma receptivity before anther dehiscence, absolutely not required. So our option is, we can clearly see, is B. that is vacuole formation is required, and asymmetric division of in the microspores. Okay, next question. Choose the incorrect statement about three-celled pollen grains. So what is this three-celled pollen grains we are talking about? The vegetative nucleus and the two male gametes, right? So they are generally long-lived. No, that is not the incorrect statement we are looking for here. Next, they are the general feature of sporophytic self-incompatible species. They are usually difficult to germinate in vitro and they exhibit high rate of respiration. The answer will be that they are usually difficult to germinate in vitro. Answer number C is the correct one. Next is, okay, RNase mediated inhibition of pollen tube leading to its inhibition in the pistil conforms to, and this question is about incompatibility. Therefore we are talking about sRNase, right, which is found in the gametophytic self incompatibility, which is responsible for disrupting the growth of the wrong or incompatible pollen tube inside the pistil, right? So number A is the correct one, and not late acting self incompatibility or sporophytic self incompatibility. No, it's in the stigmatic zone, and also RNase is not involved. There are other 3S locus alleles involved. Next is Tyler incompatibility is also not this. So gametophytic self incompatibility is the answer. Next question. Again, I think I have repeated by mistake. I have repeated this question over here again. So let's move on to the next question. A monocot root differs from a dicot root in having one of the following: a piliferous layer, radial vascular bundles, absence of endodermis, and presence of pith. Let's go and have a look at the monocot and dicot roots. So here is a picture of the monocot and dicot root. We can clearly see that endodermis is present in both of them. What is piliferous layer? It is, you can also call it the epidermis of the, from where the root hairs grow. So it is present in both dicot root and monocot root. We also know that radial vascular bundles are present in both dicot root and monocot root. The one thing which is present in the monocot root but it is absent in the dicot root is pith, clearly seen in the monocot root in the center, but we should not confuse it with the dicot stem and monocot stem where pith is present in the dicot stem, right? So yeah, so from here our answer will be the presence of pith, which is present in the monocot but absent in the dicot root. Next question. Occurrence of anthers and the stigma at different levels in the flower is referred to as A. Hardcogamy, B. Dicogamy, C. Flower constancy, and 4. Protandry. Protandry is about the timing, so it's about the timing of release, so it's not the what is asked in the question, so it's wrong. Flower constancy, no. Dicogamy, it is also no, because again that is about difference in the timing and of the female and male flower. And occurrence of anther at different level is actually hardcogamy, so our answer is number A. Next question. Malacophily refers to the pollination by snails and slugs, sphinx moths, honeybees, and spiders. It will be snails and slugs. Okay, so from here to the last question from 2014. Let's move on to 2015. Okay, so first question from 2015 is the female gametophyte in peperomia is what? They're asking the number. Is it bisporic, tetrasporic, or monosporic? Or is it 16 nucleate, 4 nucleate, 16 nucleate, or 8 nucleate? Let's just look at the chart. So here is the chart of different types of embryo sacs in Angiosperms given by after Maheshwari. So this is a very important chart that we have to memorize by heart, and look at the monosporic and examples of monosporic, bisporic, tetrasporic, and all the types of whether it is 4 nucleate, 8 nucleate, or 16 nucleate. So the one we are looking for is tetrasporic, and peperomia is actually 16 nucleate type. That is what they have asked. Is it 16 nucleate, or another option was given, is it or 4 nucleate? The answer is number C, that it is 16 nucleate and polypolar. Next question. During pseudogamous type of apomixis, polar nuclei fuse with one of the sperm cells to form endosperm. Polar nuclei fuse and initiate autonomous endosperm formation. Polar nuclei fail to fuse and endosperm is not formed. Polar nuclei never participate in the process. In pseudogamous type of apomixis, endosperm formation is mandatory. Endosperm is formed. So number C and number D gets crossed because polar nuclei does participate in pseudogamous type of apomixis. The answer is A, that is polar nuclei fuses with one of the sperm cells to form endosperm. So it is basically like fertilization, but it is pseudogamous type of apomixis, right? So that's where the name came from. Answer is number A. Next question. Callose deposition in the myocytes during the microsporogenesis mainly ensures what? Is it prevention of water loss from the myocytes, gametophytic control of the development, firmness to the microspores, or sporophytic control of development? The answer is B, gametophytic control of development. In 2015, embryology questions were a little bit very detailed. They were a little above average level questions. Next question, in an embryo sac, nucleus of the egg cell is usually located towards the micropylar and those of the synergist toward the cellulose end. B, cell wall of the egg cell is not attenuated towards the cellulose end. C, of plant species where the egg apparatus is without the synergids, the egg cell possesses the filiform apparatus and central cell invariably harbors 2 nuclei. Wrong, absolutely wrong, but our answer is number C, that in the plant species where egg apparatus is absent and it is without the synergids, the egg cell possesses the filiform apparatus. Not the cell wall of the egg is not attributed to a cellulose cell. These are not the correct answers. Our answer is C. Next question. Ubish bodies are the A, membrane bound lipoidal bodies that contribute to the egg sign formation. This is the right answer, but what are the other options? Are polysaccharide granules, membrane bound proteinaceous bodies, and callose rich bodies. We have to remember that Ubish is actually lipid. It's a lipid compound. So from here only one option had the lipoid bodies. So we could have given the correct answer from there only. So A is the correct answer. Next is one of the essential prerequisites for normal microgametogenesis to proceed is. I think they have repeated a question from 2014 itself, and we discussed as the answer is number B, that is vacuole formation and asymmetric division in the microspores. The type of cytoplasmic streaming at the region of the growing pollen tube is A, circular streaming, B, spiral, C, pulsating, and D, reverse fountain. This is fact-based question and the answer would be reverse fountain. Yes. Okay. Next question. It is again a difficult question. I mean, not so common. X bodies formed during fertilization in flowering plants represent the degenerated unfertilized male and female gametes? No. Degenerated nuclei of the synergid and the vegetative nucleus? Laggard chromosome that do not participate in syngamy, or laggard chromosome that do not participate in triple fusion? It is not a laggard chromosome, so both of this is wrong. X bodies are degenerated nuclei of the synergy and the vegetative nucleus. That forms the X body, so we should remember that. Our answer is B. Okay, so this was the last question from 2015. Let's start with 2016. Our first question is multiple embryo formation from different embryo sacs in an ovule is termed as A. False polyembryony, B. Cleavage polyembryony, C. Adventive polyembryony, and D. Gametophytic polyembryony. The cleavage polyembryony is from where we are involved with only one embryo sac, right? And adventive polyembryony, where it is formed from the different diploid cells, either the nucellus or the integument. From there the embryo sacs are formed. But false polyembryony means when more than, it is actually not polyembryony. Here many embryo sacs are present, and from there each embryo sac gets formed. Each embryo gets formed. So that's why it looks like polyembryony, but it is not. So that's why it is called false polyembryony. Our answer is number 1. Next question is an ovule which becomes curved so that the nucellus and the embryo sac lies at the right angle to the funicle is it orthotropous? No, orthotropous is complete, a straight one. Then anatropous? No, where the funiculus is actually 180-degree rotation is there. Campylotropous? No. And hemitropous is the right answer. That is it hemianatropous? It is arranged in such a manner that it goes halfway through the funicle, becomes at the right angle to the nucellus and the embryo sac. So our answer is 24A. Next question. During simultaneous types of microsporogenesis, the number of secondary spindles formed among the nuclei in the meiocyte are 2, 4, 6 and 8. The answer is 6. Next question. Which of the following statement about embryo sac and of angiosperms is correct? Nucellus of the egg cell is usually located towards the micropylar end, and those of the synergist towards the chalazal end. No. The cell wall of the egg is not attenuated. It is like we are having a same question again. It's a repeated question. This is not the right answer again. In species where the egg apparatus is without the synergids, the egg cell possesses the filiform apparatus is the right answer. The cell's central cell invariably harbors 2 nuclei is again wrong. Our answer is number C. Next question. Elaiosomes arises as an outgrowth of elaiosomes which are found in the seeds, which gives nutrition to the seed. Yeah, so that is actually an outgrowth from the funiculus. Our answer is number B. Okay, so 2016 ends here. Let's do 2017. Multiple embryo formation, but this time it's due to splitting and proliferation of the zygotic embryo is termed as what? Again, they have given the same options as last year's, say cleavage polyembryony, false polyembryony, adventive polyembryony, and secondary given over here instead of gametophyte. But our answer is cleavage polyembryony, from where it is where the zygotic embryo splits and divides into number of embryo sacs. So that is cleavage polyembryony. Next is elaiosom arises as an outgrowth of funiculus. Again a repetition. Okay, next question. Chalazosperm in cyanastrum refers to? Is it chalazal chamber formed in the halobial endosperm, or is it just its endosperm, or is it the region connecting the embryo and the endosperm and nuclear cells in the chalaza region? We know that it is the region which connects the embryo and the endosperm is called the chalazosperm. So our answer is number 3. Last question of 2017. Which one of the following arrangements of 4 nuclei in a Microspore is not observed before mitosis? Is it 1 plus 1 plus 1 plus 1? Is it 1 plus 3? Is it 2 plus 2? Or is it 2 plus 1 plus 1? Third one, that is 2 plus 2 arrangement is not seen before mitosis, which is seen after mitosis. So this option is the correct one here. Let's start with the 2018 paper. First question, is allele variants involved in conferring the self-incompatibility interact in a haplotype manner? Because it's a big question, dividing the keywords. S allele variants in self-incompatibility, we are talking about the S locus, which are responsible for the self-incompatibility factor. And what is haplotype manner? What is meant by haplotype is when a number of genes are from a same parent is inherited together of an organism is inherited together from a single parent is haplotype. I'm not writing it here. Okay, so this S locus which is interacting in a haplotype manner, because is it S allele products are expressed only after the completion of meiosis in germline cells. It is true, but it is not the reason why it's haplotype. Next, only then the allelic variability could be generated. No, this is wrong. It is an essential requirement for the success of gametophytic self-compatibility system. It is important for GSI, but that does not. It is not the reason behind the haplotype manner. Last option is S locus with male and female determinants is inherited together as one unit. This is our answer. That is why S alleles are interact in a haplotype manner. The answer is given itself in the question only. We just have to know the definition properly. Next, in gametophytic self-incompatibility GSI system, which of the options are correct? Outcome of the interaction is actually determined by the genotype of the pollen producing parent. No, this is a no. Next is pollen grains are 3-celled. That is not the factor in GSI. Next is S locus products are synthesized after completion of meiosis, and growth of the pollen tube is arrested in the stigmatic zone. No, in GSI it is not arrested in the stigmatic zone. It is actually in the pistil zone by RNAs. So our answer is actually the products are synthesized after the completion of meiosis. So 23 is the correct answer. Next question. Nuclear endosperm formation is characterized by nuclear endosperm formation. Is it nuclear divisions accompanied by cell wall formation? No, cell wall formation is not involved in nuclear division. Next, so this is wrong. Primary endosperm nucleus dividing to form a large micropylar chamber and a small chalazal chamber, that is halobial. So this is not nuclear again. Next is autonomous development of endosperm without fertilization, apogamy. So this is not nuclear endosperm formation. Free nuclear divisions not accompanied by cell wall formation is the correct definition of nuclear endosperm formation. So this is our answer number 4. Here we have come to the end of all the previous year questions, except 2019 is missing over here, but this gives an idea of what the type of questions come for embryology, or you can also call it the reproductive biology of angiosperms. So okay, I hope this was helpful for you. Thank you for watching.